Okay, good morning, everyone. It's nice to see everyone in the virtual world today. I hope you're doing well and at least we have some sunshine outside. Some big stuff happening. We have our third lab exam today, so please be sure that you sign in to Class Marker and take your final lab exam of the semester. So, once that's all over, hopefully you'll feel a sense of relief that you've finished that portion of the course. And of course, congratulations on making it to your final week of lecture, right? We have our last two lectures of the semester this week, and then your final exam is on Monday, May 11th, okay, which again, you'll have from 7 a.m. to midnight on Monday the 11th to take the final exam. So lots of things going on right now. Do your best to stay focused for the next week so that you can finish the semester strong. Do you have any questions at this time? Okay, not seeing anything. So we'll begin the final chapter of the semester, folks, okay? So we're going to go to chapter 16 at this time, which starts on page 548 of your textbook. Let me move us into our normal Microsoft Word document here. Okay, so at this moment, folks, you should be seeing a chat window and a Microsoft Word document entitled Biology 121 Lecture Chapter 16. Okay, so folks, the title of this chapter is Sensory, Motor, and Integrative Systems. So we're going to be providing a lot of information about these three major topics over the next 2 days. So let's begin. I'm going to start with some basic definitions. Mosena, we did not, Mosena, I see your question on the chat window. Let me see something, one moment. Oh, yes, okay. Please, Mosena, can you type into the chat window what the last line of your notes from last week was? Okay, hold on one moment, folks. Let me bring up the notes from last time to see where we did finish. Okay. Let's see, the date on that would have been the 30th or the 29th. Okay, let's see here. I see, nicotinic receptors. Okay, thank you, Messina. Yes, I suppose we probably should finish that. Okay, folks, so let's take maybe 10 minutes here and finish the notes on the autonomic nervous system. Okay. All right, so I'm going to keep that document on top of your screen at this point, and this is a continuation of what we did last time. Let me put this content into our new notebook here, and I will stick it up here. Okay, so this is from last time. So we will finish chapter 15 first. So this is chapters 15 and 16. Okay. Okay. All right, thank you for reminding me. Okay, let's continue here. So we did talk about the nicotinic receptors. All right, I have a few additional comments to make about those receptors. And so, okay, the nicotinic receptors are most commonly found at the neuromuscular junctions, at the chromaffin cells of the adrenal medulla. Okay. Now, the activation of the nicotinic receptors by acetylcholine, as you know, causes depolarization and excitation of the postsynaptic cell. Once again, activation of nicotinic receptors by acetylcholine causes depolarization and excitation of the postsynaptic cell. Now remember, we're talking about the nicotinic receptors, and this is in the broader category of cholinergic neurons. Okay, cholinergic neurons have two different kinds of receptors. Okay, nicotinic receptors and muscarinic receptors. Okay, so before I continue, do you have any questions at this time? Okay, let's continue here. All right, so we talked about the nicotinic receptors. Now we need to get some notes into you about the muscarinic receptors. Okay, muscarinic receptors are named because of actually a mushroom poison called muscarine. Muscarine is an agonist, meaning it binds to and activates the same receptors as acetylcholine.
Dr. Allen, I have one question.
Okay.
What do you mean? Is it like the nicotinic receptors? Is that like for, like, what does that actually mean? Is it like for nicotine? Like, it sounds like something from like nicotine addiction or something.
Okay, so nicotinic receptors are found in the plasma membranes of cholinergic neurons. But
Okay.
the reason they are called nicotinic receptors is because scientists noted that nicotine, the same thing that you have in cigarettes, nicotine as a molecule is able to bind and activate these same receptors that acetylcholine can. So that's how they got their name.
Oh, okay. Okay, that makes more sense.
Okay. All right. So in a similar fashion, the muscarinic receptors are a second type of receptor that are found within the plasma membrane of cholinergic neurons, and they're named muscarinic after the chemical that activates them. Muscarine is the chemical normally found in mushrooms that can activate them. So muscarine and acetylcholine are both capable of binding to and activating these receptors. So as I complete my second thought here, muscarine is an agonist of acetylcholine at these receptors, okay? So when we say we've identified an agonist, we mean we've identified a molecule that can activate a receptor, that can bind to and change the shape of that receptor. And depending on the nature of the shape change, we will either activate it or turn it off, okay? So nicotinic receptors are bound by and activated by not only acetylcholine, but nicotine. Muscarinic receptors are bound by and activated not only by acetylcholine and muscarine, okay? Now, of course, we get different responses, right? Nicotinic receptors will bind acetylcholine, and so will muscarinic receptors, but you'll get a different physiological response because while they are both binding acetylcholine, they both can attach to it, they respond with a different shape change, so you get a different physiological response. Okay, let's continue here. Muscarinic receptors are commonly found in smooth muscle tissue, cardiac muscle, and glands. Okay, and completing this topic, activation of muscarinic receptors can be depolarizing or hyperpolarizing, okay? And the reason for that is, so we have two different broad categories of cholinergic receptors. They are respectively nicotinic receptors that I'm highlighting, and then muscarinic receptors. But within the category of muscarinic receptors, there are at least 5 different classes of muscarinic receptors. There's 5 different kinds of muscarinic receptors, which explains why we can get a variety of different responses to the same molecule. Okay, if you want visualization, I mean, it's not really a picture, but it's more of a table. If you want a table for this, have a look at page 539 of your textbook. Okay. Here we have different types of receptors. The top part of the table has the cholinergic receptors, nicotinic and muscarinic. Okay. All right. So let's see here. Activation of the muscarinic receptors can be depolarizing, which is to say it's going to be excitatory, right? If we depolarize a cell, we electrically excite it, or hyperpolarizing. If we hyperpolarize a cell, that is an electrically inhibiting stimulus. So that's inhibitory. Okay, depending on the type of muscarinic receptor. Okay, and I'll just put in parentheses here that medical science is aware. Medical scientists have over the years documented at least 5 different flavors, 5 different versions, 5 different types, 5 different kinds, 5 different variations of muscarinic receptors. The technical term in biology are 5 different isoforms. At least 5 different isoforms, which meaning 5 different versions of muscarinic receptors have been identified by medical science. Okay. All right. So as with all things, the story gets ever deeper, ever more complicated. But that's as deep as we're going to go. Okay. Please note, I know this is a lot and it can be confusing, and really part of your battle, as it's been the entire semester, is making sure that you clarify how information is organized in your own brain, right? Nicotinic receptors are one type of cholinergic receptor. They bind acetylcholine. but they're named nicotinic because they also bind nicotine. Muscarinic receptors are the second different kind of cholinergic receptor. They also bind acetylcholine, okay? But they do so in a different way than the nicotinic receptors. Within the muscarinic category, there are 5 different forms of it. And depending on the form of muscarinic receptor, you can either get an excitatory stimulus or inhibitory stimulus. Please know that nicotine and muscarine do not activate each other's receptors, okay? They do not activate each other's receptors. They are specific for their own specific type of receptor. Okay. All right, so that's some good warm-up exercise here to get our thinking caps on, so to speak. So any questions right now? Okay, so remember from last time, the broader conversation we're talking about right now is the autonomic nervous system, the neurotransmitters we find that are at work in the autonomic nervous system, and the receptors that bind to those neurotransmitters. Okay, so those are the cholinergic neurons that you see at the top of table 15.2 on page 539. Finishing this topic out so we can get to chapter 16, we've also got to catalog the adrenergic neurons and the adrenergic receptors. So adrenergic, I can't even say that, adrenergic neurons release norepinephrine, which is sometimes also called noradrenaline. Okay, basically what you think of as adrenaline during the fight or flight response. Okay, so these adrenergic neurons contain adrenergic receptors which bind 2 different substances. They bind norepinephrine and this epinephrine, which are chemical cousins of each other. Okay, and I'll give you a little bit more about each of these. Norepinephrine is released as a neurotransmitter. By sympathetic postganglionic neurons and it's a big or here. Norepinephrine is released as a neurotransmitter by sympathetic postganglionic neurons or as a hormone released from the chromaffin cells of the adrenal medulla. Okay, so that highlights an example of how the same substance in one context can be a neurotransmitter, and in a second alternative context can be a hormone. Okay, right? It's the same molecule. Norepinephrine is the same whether it's released by the neuron or it's released by the cell of the adrenal gland. It's the same actual molecule. But when it's released from the neuron, we describe it as a neurotransmitter. When it's released from the adrenal cells, it's described as a hormone because it gets access to your bloodstream. Okay, so don't let these words such as neurotransmitter and hormone throw you for a loop because they're simply molecules that convey information. They are signaling molecules. That's all they are, is they are signaling molecules to communicate information from one cell to another cell. That's all they are. Okay. Questions? Okay, now epinephrine is released primarily as a hormone by the chromaffin cells. But it has been detected as a product that is released by neurons in the medulla oblongata of the brain. Now, when we say it has been detected, that should tell you that it's been detected, but we don't really understand it very much. In other words, we are reaching the limit of human knowledge and understanding at this moment in time. Yes, epinephrine has been detected as being released by cells in the brain, but we don't know exactly what its actual molecular role is in the brain. But it has also been detected as a released product by neurons in the medulla oblongata of the brain. Okay, so let's be honest. Both norepinephrine and epinephrine can be neurotransmitters, and they can be hormones, depending on the context of what kind of cell is releasing the molecule. Okay, all right. Now, as your book shows you on page 539, there are two main types of adrenergic receptors, the so-called alpha and beta versions. Okay, alpha and beta. And speaking in generalities at this time, because we're not studying any specific circumstance anymore. Generally, activation of alpha 1 and beta 1 receptors leads to excitation. Okay. And generally speaking, activation of alpha 2 and beta 2 receptors leads to inhibition. Okay. Okay. All right. So, folks, are there any points about this that you need clarity on? Do you have any questions that I can help you with? I think this is where I'm going to wrap up the topic and let it rest. So are there any questions you have about this before we move on to chapter 16? Okay. Not hearing or seeing anything from you, I will assume that you're okay. And let's start chapter 16 here then. Okay. So chapter 16. Okay, Ashley, can receptors like alpha 1 and beta 2 be activated at the same time, even though they cause different things? Yes, Ashley, they can, depending on what kind of cells we find them in. We could have the same substance, norepinephrine, binding to two different kinds of receptors on two different cells at the same time. Yes, that definitely happens. Okay, other questions? Okay, all right. Okay, chapter 16 then. All right, now, chapter 16, sensory, motor, and integrative systems. Let me begin this chapter with some opening definitions. Okay, so we need to talk about sensation. A sensation refers to the conscious or subconscious awareness of changes in the external or internal environment. So, you know, when cold air breezes up, when cold air pushes across your body, that's a sensation. Or it could be that you have an unusual reflex in your esophagus and you start getting some heartburn. That's a sensation as well. Okay, so both of those are sensations. Okay, now the sensation can be conscious or subconscious, right? If you remember the old idea of subliminal messaging that, you know, actually is illegal today, you're not allowed to have subliminal messaging in commercials these days. That's illegal, but that's also a possibility, right? Your body is sometimes aware of things that you're subconsciously aware of that you're not aware of at the conscious level of waking. Okay, so sensation is the conscious or subconscious awareness of changes in the external or internal environment. Okay. Number two, perception. Okay, perception is specifically the conscious awareness of a sensation. Okay, so we have to be very careful here when we throw out, at least for purposes of understanding this concept, when we throw out the word perception, we're saying that you are consciously aware of something happening. Okay? But when we throw out the word sensation, it means your body is aware of something that's happening, but you might not be consciously aware of it. You know, if you think about it, you know, every year people end up having heart attacks. And so heart attacks can be caused by a number of things, but certainly one thing that can cause a heart attack is a blocked artery. Well, if you think about it, usually you don't feel an artery getting blocked, right? You don't feel it. You're not consciously aware of it. Now, your brain might be aware of it subconsciously, and there might be changes in how your body physiology is functioning. There might be changes in your blood chemistry. All kinds of things might be changing, but you're not actually aware of it consciously. You don't think about it throughout the day. And that's why when people end up having a heart attack, it's such a shock. It's like, I had no idea I was sick, or I had no idea that Uncle John was sick, right? Because you're not consciously aware of many of the things actually happening inside your body. Okay, so next idea is to introduce this idea of sensory modalities. Okay, this is a topic on page 549 of your textbook. Okay, sensory modalities refer to the types of sensations that we can experience, that your body can experience either consciously or subconsciously. This phrase refers to the types of sensations that we can experience. Now, there's two broad categories here, right? There's general senses. This would include the somatic and visceral senses. And when I say somatic senses, the term somatic means body, but somatic senses are typically things that happen at the surface of your body, such as you feel an itch on your skin, or you feel the warmth of water as you're, swimming in a heated pool or something like that. And then there are the visceral senses, which are things like your arteries stretching and things like that, which you're not typically aware of, okay? So we have general senses, which include the somatic and visceral senses. All right, let's talk about somatic first. Somatic senses, these are sensations such as touch. So when you touch an object, pressure, you think about somebody gives you a bear hug. Vibration, maybe you're trimming your yard with a weed whacker and you feel the vibration of the weed whacker as it is running. Itch, again, we mentioned itch, the idea that you feel an itch on the surface of your skin. Tickle is also a general sense. So, when you tickle a baby or when you are tickled and you giggle. Warmth, coldness, pain. Pain is a somatic sense, which is a type of general sense, okay? Pain is a general sensation. Pain and proprioception, okay? Big fancy word here. Proprioception refers to your brain's awareness of where its body parts are in three-dimensional space. So as you sit at your desk typing in your notes, you don't have to look at your right foot. You don't have to look at it, but yet you can detect where it is, right? You have a sense of where your right foot is without actually looking at it. That is proprioception. Okay? All right, so these are the somatic senses. In addition to that, the second form of general sense are the visceral senses. These senses would include the following sensations. Pressure, okay? So, some forms of pressure you will be consciously aware of and others you won't. For example, when your urinary bladder is full, right, it's going to stretch the wall of the bladder because there's going to be increased pressure in the bladder by too much urine volume that you're aware of. And then oh, I have to go pee. I have to go to the bathroom. Okay? But then there's other different visceral pressure sensations that you will never be aware of, such as a blood vessel stretching. Okay, so even pressure, there's different versions of pressure. All right, so sensations such as pressure, stretch, chemical detection, right? Chemical detection, such as how much carbon dioxide is in your bloodstream. Nausea, hunger, and temperature, okay? Your organs, the inside viscera, they can detect abnormalities in temperature as well. Okay, so we have the general senses, which include the somatic senses, which we've defined, and the visceral senses, which we have defined. And then there are the special senses, things that we probably think of more appropriately when we hear the word sense, like a sense of taste and things like this. Okay, so these are sensations such as smell, taste, vision, hearing, and balance. So when we talk about sensory modalities, we first break it into two categories: general senses and special senses. Within the general senses, we have somatic senses, which typically happen roughly at the surface of your body. And then we have the visceral senses, which happen internal with your organs. Then we also have the special senses, which we spend a lot of time thinking about: hearing, taste, smell, vision, and whether you're going to fall over or not, your sense of balance. So please keep in mind that no matter what kind of sensory modality we are discussing, please understand that an individual sensory neuron can only transmit information for a single sensory modality. Okay, you cannot have a single neuron being part of a general sense and a special sense at the same time. That never happens. That doesn't work that way. Okay. No matter what kind of sensory modality we are discussing, understand that a single sensory neuron can only transmit information for a single sensory modality. It cannot do multiple. It cannot multitask, right? Multitasking is something that was kind of trendy years ago, saying that everybody can multitask. Well, neurons do not multitask in terms of sensory modality. Okay, a single neuron can only transmit information for a single sensory modality. Okay. All right, folks, any questions right now? Okay, so let's talk about how sensation actually occurs. And I'm going to be talking about the 4 events that you see on page 549. How does sensation occur? Remember, we're talking about the conscious or subconscious awareness of a change in the internal or external environment. So how does a sensation occur? There are 4 events that must happen. Okay. And they are: number 1, stimulation of a sensory receptor. Okay. Remember that a sensory receptor is a neuron or a specialized cell that communicates to a neuron. Okay. When we say sensory receptor, the neuron, or in some cases the specialized cell that communicates with the neuron, can be part of a larger group of cells that forms a structure. So the one that's immediately coming to mind is a taste bud. Okay. The neuron, and sometimes specialized cell, can be part of a larger group of neurons that form a structure, like a taste bud. So let me see here. Okay. If you take a look on page 582 of your textbook, so here we're getting into chapter 17, but page 582, you see an example of what a taste bud actually looks like on the surface of your tongue there. Okay. And so take notice that a taste bud, as we talk about it, is not an individual cell, but it's a group of cells. If you look at page 582, picture B is showing you where the taste bud is actually embedded in the tissue of the tongue. And then you go down to picture C, you see a blown up diagram of a taste bud. You can see several things, right? You can see gustatory receptor cells, you can see basal cells, and you can see first order taste neurons. Okay, you also see supporting cells. All of these cells, the supporting cells, the gustatory receptor cells, the basal cells, and the first order taste neurons are collectively part of the entire structure that we call a taste bud. And that entire collective structure that we call a taste bud is a sensory receptor. Okay, so a sensory receptor can be an individual cell, but it can also be a group of cells that are working together for a common goal, which in this case, as a taste bud, is to detect molecules in the food that you're putting in your mouth. Okay, so let me just put down figure 17.3 there in your notes if you want to look at it at a later date. Okay. Okay, questions right now? All right, so I have a question for you. Okay, if you look on page 550, you see figure 16.1 there, figure 16.1. And so here we have different types of sensory receptors. Okay, so my question for you here is to be sure that we're not just putting new information into your notes, but we're starting to integrate and see the connectedness between different topics we've talked about here over the past several chapters. So let me ask you a question coming through the chat window on figure 16.1. What is another name for a receptor potential on figure 16.1? You see that blue ball there, and it's labeled receptor potential. Okay, on picture A and C, receptor potential. What's another name for a receptor potential? What do you think, gals? We have talked about this. I promise you we've talked about it. I just want to be sure you're picking up, you're getting it into that brain. Okay, thank you very much. That's exactly what I'm fishing for. It is a graded potential. That's exactly right. Nicely done, Ashley and Pam. Receptor potentials are the same thing as a graded potential. A receptor potential is going to be the same thing as a graded potential, okay? All right, so when you get a bear hug, somebody squeezes your body, when your neurons embedded in the dermal layer of your skin, when they activate, the very first thing they're going to get is a graded potential, right? If it's strong enough, you might generate an action potential that then travels into the spinal cord and up to the brain so that you have awareness that somebody's giving you a bear hug. But it all starts with a stimulus which depolarizes neurons embedded in the skin, and it all starts with a graded potential. Okay, nicely done. Okay, so second event that has to occur with the sensation is transduction of the stimulus. Okay, transduction as a word is kind of a fancy word. It basically means to convert, okay? Means to convert. For example, when photons of light, okay, when light, which is, is made of photons, if you've had a basic physics course, when light strikes the surface of your retina, where your photoreceptors are embedded, okay? And the retina is at the back of your eyeball. I know we haven't lectured on it, but there has been some study on the structure of the anatomy of the eye for today's lab exam, so you should know what the retina is, okay? When photons of light strike the photoreceptors embedded in your retina, the information contained in the light, okay? Information contained in the light. And you might ask yourself, well, what information is contained in the light? Well, the intensity of the light and the wavelength of the light. In other words, what color are we receiving? Are we receiving red wavelengths of light? Are we receiving yellow wavelengths of light? Are we receiving blue wavelengths of light, green? What might it be? That's the information I'm talking about. The information contained in the light is converted into the electrical signal of a graded potential. Okay, do you see that a stimulus always, when a stimulus activates a sensory cell of one form or another, it always starts in the sensory cell as a graded potential, which then, if it's strong enough, if that electrical agitation of the graded potential is strong enough to reach threshold at the axon hillock of the axon, then we generate an ax potential that goes into the central nervous system. Okay, let me read my notes here. For example, when photons of light strike the photoreceptors embedded in your retina, the information contained in the light is converted into the electrical signal of a graded potential. Does that make sense, folks? There's information in the light that has to be converted into an electrical signal, okay? If we're talking about sound waves, right? We're talking about sound waves. The sound waves of my voice, as they're being transmitted through your computer speaker, are striking your eardrum, and it's causing the eardrum to mechanically vibrate. So the information of the sound waves is being transduced into a mechanical vibration in your eardrum, which is then mechanically moving three little bones behind the eardrum, which is then mechanically sloshing the liquid inside your cochlea, which then is used to move tiny little hair cells inside your cochlea, which then depolarize neurons inside your inner ear with a graded potential, which will then summate at the axon hillock for an action potential, which will then go up the vestibular cochlear nerve into the brain. And you say, oh, I hear Dr. Allen talking about some nonsense. Okay? This is how this all works, gang. Transduction is the conversion of information from one format to another. Okay. Question coming through the chat window. Does transduction make sense? Okay, all right, great. Okay, please let me know if you have questions. Feel free to interrupt me, that is okay. All right, so the third event that must occur here is the generation of the nerve impulses. I kind of got ahead of myself with my examples here. Okay, if the graded potentials are strong enough, threshold, which we mentioned before, is typically on your average neuron is about minus 55 millivolts, okay, on average. Threshold is reached and an action potential is created. Okay, neurons that transmit nerve impulses from the peripheral nervous system into the central nervous system are given a name, and they are called first-order neurons. Okay, neurons that transmit nerve impulses from the PNS, the peripheral nervous system, into the CNS, the central nervous system, are called first-order neurons. Okay. So you noticed, I won't ask you to go back to chapter 17, but you notice at the basal area of the taste bud, there was something called a first-order neuron. That's because you're transmitting information from the peripheral nervous system, in that particular case, tissue embedded in the flesh of your tongue, into the brain and spinal cord. Okay. Okay. Any questions? Okay, number 4. The final thing is integration. Okay, we're talking about what steps are involved, what events must occur during a sensation. Integration of the sensory input. Okay, a particular region of the central nervous system receives and processes, in other words, integrates the sensory nerve impulse. Okay, a particular region of the central nervous system receives and processes, integrates the sensory nerve impulse. Okay, if we continue with our eye example that we've been talking about here recently, the action potential is sent along the optic nerve into the occipital lobe of the cerebral cortex so that you can perceive the flower that you're looking at, or the giraffe at the zoo that you're touring, or whatever it might be. Continuing with our eye example, the action potential is sent along the optic nerve into the occipital lobe of the cerebral cortex. So you can see the giraffe at the zoo. Okay? All right, questions so far? This darn class goes way too fast to get everything done. Okay, let's continue. All right, let's talk some more about the somatic senses. Okay, we have defined them. Let's talk a little bit more about the detail of the somatic senses. Okay. Okay. Somatic sensations here. Somatic sensations. Okay, so we recall that there's basically 4 types of somatic sensations. They are tactile, right, where you touch and you feel something, the roughness of a surface. Thermal, heat, or the absence of the heat, coldness. Pain, pain would be involved if you are physically damaging tissue in some way, you're going to get a pain sensation. And proprioceptive. Okay. Now, the tactile, thermal, and pain sensations result from stimulation of receptors embedded in the epidermis, dermis, or hypodermis. And sometimes in the case of pain, I mean, you can get pain because, you could get pain in your chest because there's something going on with your heart. So in other words, you can get organ-based pain or so-called visceral pain. Tactile, thermal, and pain sensations result from stimulation of receptors embedded in the epidermis, dermis, or hypodermis, and sometimes, in the case of pain, in the viscera itself. Now, by viscera, I'm referring to the organs. So, there are actually, let's see here, I'm looking at table 16.2 at this point. Table 16.2, page 557. There's 10 different types of receptors for somatic sensations. And we're just gonna we're gonna catalog these down in your notes. Okay. 10 receptor types, table 16.2. All right, we have Roman numeral little I, corpuscles of touch. Corpuscles of touch detect the onset, detect the beginning of touch, and low frequency vibrations. If something's vibrating, you're going to activate a corpuscle of touch. Trying to spell that properly for you. There we go, corpuscles of touch. Okay. These detect the onset of touch and low frequency vibrations. Okay. Now, corpuscles of touch, they're often encapsulated. Okay, let's see here. Yeah, take a look. If you take a look on page 553, you can see figure 16.10. And I don't, hopefully this image that you see in figure 16.2 is at least a little bit familiar to you from when we talked about the structure of your skin way back in a previous chapter. But do you notice that the corpuscles of touch are typically embedded there in the epidermal ridges there of your skin? Okay, so those are the corpuscles of touch. Next, we have the second type of receptor, which is the hair root plexuses. Okay, these are found in areas of your skin that have hair, and they allow you to consciously be aware of the motion of your body hair. Okay, they allow you to perceive the motion of body hair. Okay. We have, as well as that, we have type 1 cutaneous mechanoreceptors. Okay, when you hear mechanoreceptor, think some sort of mechanical motion of the tissue. So we're talking about typically sensations involving stimuli that stretch the tissue or create some sort of pressure on the tissue. Type 1 cutaneous mechanoreceptors. Okay, these are senses. These sense continuous touch. Okay, so when someone's holding your hand and they hold your hand for 2 minutes, that sense of continuous touch is going to activate type 1 cutaneous mechanoreceptors, as well as the pressure of someone gently squeezing your hand as they hold your hand. Okay? In addition to that, we have type 2 cutaneous mechanoreceptors. It just goes to show how complicated the anatomy of your body has to be to allow you the luxury of all these wonderful sensations that life can give us. Right? Think about that wonderful sensation of giving your child a hug. So type 2 cutaneous mechanoreceptors senses skin stretching and pressure. All right, so that's definitely going to be involved with some forms of a bear hug or something like that. Then we have the 5th form of receptor type. This is the lamellated corpuscles. Okay, these sense high frequency vibrations. Right. So anything that reaches a frequency level that's greater than what we would typically perceive with the corpuscle of touch, that's going to activate the lamellated corpuscles. And then we mentioned up here that there are 10 receptor types. So we've documented 5 of them so far. The 6th version are free nerve endings. And within that category, there's 5 types of them. So category 6, okay, category 6 here is free nerve endings, which come in 5 varieties. Okay, so free nerve endings allow you to perceive pain, itch, tickle, cold, and warmth. And then we have 5 different kinds of neurons that are customized to allow you to detect these sensations. Pain, itch, tickle, cold, and warmth. Okay, those are your 5 right there. All right, a couple other notes for you here. Pain receptors are also called nociceptors. Okay, that's a strange word, and I'll spell it for you. Pain receptors are also called nociceptors. That's nociceptors. Okay, these are activated. These pain or nociceptors are activated whenever a stimulus is actually strong enough to start structurally damaging tissue. Okay, that's their job. Is Mother Nature created them so that if a stimulus is so intense that it might physically harm your body, that's when the pain receptors activate. Okay. Fast pain is precisely located to the area of the body that's receiving the stimulus, creating the pain. But slow pain tends to spread out. Okay. It tends to be more of a diffuse nature. Okay, so think about a toothache. I mean, with a toothache, you know, you can really feel the pain right at the location where that tooth is irritated. Okay, but given enough time, the pain from your tooth might spread out through your entire jaw area. Okay, so the fast pain would be the pain right at the irritated tooth, and the pain that you're feeling over the general uncomfortableness of your jaw is going to be the slow pain. Okay, fast pain is precisely located to the stimulated area, but slow pain tends to be more spread out. Okay. Now there's another aspect to pain. I mean, you know, none of us like pain, but it is our body's way of making us aware that something's wrong, and if anything can reasonably done to correct the problem, we should probably do it, right? So even though it's annoying to experience pain, it's meant to be a protective thing so that we can try to take corrective action. In addition to these aspects of pain that we've just put into your notes, there's also the idea of visceral pain. Okay, it's the visceral pain. You know, think about, God forbid, think about pain with your heart because it's experiencing some kind of trauma. It might be on the verge of a heart attack, and you might experience referred pain, which goes up your arm or, you know, up into your shoulder and neck area, actually. For some folks, people that are going to get a heart attack, they actually develop nausea in the stomach. Okay, so you get this idea of pain that's being experienced but far away from the actual problem site. With visceral pain, it is common to also experience referred pain. Okay, so we have some catchphrases in here, right? Fast pain is a catchphrase. Slow pain is a catchphrase. We have visceral pain. And we have referred pain. Okay. This occurs in the case of referred pain. It happens because the area stimulated and the area that's not directly stimulated are controlled by the same region of your spinal cord. That's the commonality here. If you have visceral pain at one location and you have referred pain at a different location, okay, you can bet there's a commonality with the nerve fibers as they leave the spinal cord. With visceral pain, it is common to experience referred pain because the area stimulated and the area not directly stimulated are controlled by the same region of the spinal cord. Okay? All right. Okay. So ladies, that takes care of note-taking for the somatic sensations. Before I continue into the proprioceptive sensations, do you have questions? I know this is a lot to process. This is very intense, right? I mean, we could make a semester-long course just on the nervous system. There's no question that we could do that if we were so motivated to do that. Okay. All right, so we've talked about the 10 receptor types, 1 through 6, and number 6 has 5 varieties. So 10 receptor types of the somatic sensations. Okay, now we're going to talk about the proprioceptive sensations. Remember, proprioception is your body's awareness, your brain's awareness of where body parts are in three-dimensional space. You know where your foot is at this time without looking at it. Proprioceptive sensations. Okay, so there are 3 receptor types for this. Okay, and let's see here. I'm continuing to look at table 16.2 for this. Okay, so you can use table 16.2 for quite a lot. And, you know, just be aware of some of these tables for your final exam, because, you know, you might have to look up a small little factoid to answer a question. And these tables can be very helpful to get information rapidly, okay? So there are 3 receptor types. And I'm talking about table 16.2 here. There are muscle spindles, tendon organs, and things called joint kinesthetic receptors. Muscle spindles, tendon organs, and joint kinesthetic receptors are receptors that allow you to receive proprioceptive sensations. We're going to talk about each of these in turn. Okay, so first we have the muscle spindles. Okay, the muscle spindles you can see in figure 16.4, page 556. Figure 16.4, page 556. These receptors monitor changes in the length of the skeletal muscle, and they also participate in the stretch reflexes of the muscle. So they monitor changes in the length of skeletal muscle and participate in stretch reflexes. Okay. So when you look at figure 16.4, let me point out a couple features that, you know, I hope that you're thinking about as you study. Okay. So we're looking at figure 16.4. Okay. So folks, you all know where, you know, the biceps brachii is, right? You all had to know that for lab exam 2. You really are expert at this anatomy, more so than the average Joe on the street. You have accomplished a lot this semester. You've worked very hard. So you know where the biceps brachii muscle is. And my point is here, notice how the muscle tissue is actually made of 2 different kinds of muscle cells, the so-called extrafusal and intrafusal. Okay, if you're looking at figure 16.4, take a look at the bottom of the picture itself. Do you see the phrases extrafusal and intrafusal? Okay. All right. So, Pam, what reflexes you're talking about? Oh, okay. Hold on a sec. Okay. By stretch reflexes, I mean, you know, if you've ever been at the gym and you've stretched your muscles out before doing a sporting activity of some kind, right? You stretch the muscle out, and then their reflex is to naturally contract back to their normal length. That's what I'm talking about. You stretch the muscle out, then the reflex is to go, once you finish the stretch, the muscles go back to their normal length. Okay? Any other questions? Okay, so let me document this extrafusal and intrafusal muscle cell types. Note how the biceps brachii muscle, and you know, biceps brachii is just an example. This could be true of any skeletal muscle. Is made of two different types of cells, extrafusal and intrafusal. Okay, let's define and contrast these. Extrafusal muscle fibers. I see the time, folks. Hang with me for like a minute or two here, and we'll get this subtopic wrapped up. Extrafusal muscle fibers are the normal contractile types of muscle cells that you learned about in chapter 11 when we talked about skeletal muscle contraction, you know, where we talked about the calcium triggering activation of the sliding filament model of muscle contraction. This is the extrafusal muscle fibers. Okay, these are the normal contractile type of cells you learned about in chapter 11 this semester. And then we have the intrafusal muscle fibers, which are, they anatomically speaking, the intrafusal are deep. They are deep to the extrafusal cells. And these are the actual muscle spindle receptors. The muscle spindle receptors are made of the intrafusal muscle cells, the intrafusal muscle fibers. Okay. Okay, let's highlight that. The intrafusal muscle fibers are the actual muscle spindle receptors. They are the actual muscle spindle receptors. And what that means is that these cells report to the central nervous system how much stretch a muscle is experiencing. Okay? Okay? These cells report to the central nervous system just how much stretch a muscle is experiencing. Okay? If the muscle is experiencing too much stress, we might have to engage more extrafusal fibers to prevent an overstretching and damaging of the muscle itself. And if there's not enough extrafusal fibers to do that, we might have to tell our skeletal system to relax its grip on something so we don't damage our muscles. The information from the intrafusal fibers helps the cerebellum coordinate muscle contraction. The information from the intrafusal fibers helps the cerebellum, which at this point you know exactly where that's at because you have your exam today, coordinate muscle contraction. Okay. All right. So let's see here. I'm looking at, oh, wow. Okay. We're still going to have a pretty full day on Wednesday. There's still so much to talk about. But at any rate, this is a good stopping point. Ladies, do you have any questions right now? Okay, I hope you're okay. I know this is intense. I would never say it isn't. This is very involved. But take heart, you are close to the end. So grin and bear it, dig deep, Stay focused, organize your information, organize your notes so that when you have to take your final exam on Tuesday, anything you can't answer, you can quickly look it up in your book and notes. Okay, that's your game plan for success. Once again, do you have any questions before we stop for today? Okay, I'm not hearing anything and I'm not seeing anything on the chat window. So I'm going to stop sharing my screen. I will send the notes to you later today. Okay. So do you have any questions before we take a break? You know, today you have to do the lab exam, so there's really no lab after today's lecture here. There's no online component to it. But is there anything I can help you with before that, before your lab exam or anything?
Are we going over chapter 21
Chapter 21 homework?
for lab? Yeah.
I can do that if you would like it. I have no problem doing that.
Okay.
All right. Okay. All right. So folks, if I don't see you, good luck with your lab exam. And, you know, I will be monitoring email. So if you have any technical problems, shoot me an email and we'll work to get it fixed. We'll work to get it fixed. Otherwise, I'll see you on Wednesday. I'm going to take about a 10-minute break here. I'll be back online for anybody that does want the answers to the lab homework. We can do that. So see you, folks. Good luck on your test today. See you later. Okay, Ashley and Suzette, we'll take a look at your exercise 21 homework, okay? So please have open page 339 of your lab manual, reviewing your knowledge. Letter A, cranial nerve identification. Here we go. Number 1 is cranial nerve 6, which is Roman numeral VI. Number 2 is Roman numeral XI, which would be 11. 3 is Roman numeral VII, so that would be 7. Number 4 is Roman numeral IX, which is cranial nerve 9. Number 5 is Roman numeral XII, which is cranial nerve 12. Number 6 is Roman numeral III, so 3. Number 7 is Roman numeral I, so that's 1. Number 8 is Roman numeral II, so that's cranial nerve 2. Number 9 is Roman numeral V for 5. Question 10 is Roman numeral IV, which is 4. Question 11 is X, which is cranial nerve 10. And 12 is VIII, which is 8, cranial nerve 8. Do you need any of those repeated? Okay, let's take a look at the next group of 12 by writing in the name. Number 1 is the olfactory nerve. 2 is the optic nerve. 3 is the oculomotor nerve. 4 is the trochlear nerve. 5 is the trigeminal nerve. 6 is the abducens nerve. 7 is facial. 8 is vestibulocochlear. 9 is glossopharyngeal. 10 is the vagus nerve. 11 is the accessory nerve. 12 is the hypoglossal nerve. So some really big words in there. Do you need any of those repeated?
I have one question. For the olfactory, if you put bulb, will you still get credit on it if it's on a test?
Could you repeat that for me, Suzette?
The olfactory, if you put bulb at the end, would you still get credit for it? Or does it have to be nerve?
Well, I would probably give you credit for that. The bulb is simply referring to a bunch of neurons as a group, so that would be okay.
Okay.
All right. Let's go to page 340. Letter B is the cranial nerve function. Okay, number 1 is S for sensory. 2 is also sensory. 3 is M for motor. 4 is motor. 5 is B for both. B for both. 6 is motor. 7 is both. 8 is sensory. 9 is both. 10 is both. 11 is motor. And 12 is motor. Okay. Letter C, cranial nerve action matching. Here we go. Okay, number 1 there is the letter E. 2 is the letter H. 3 is the letter L. 4 is F. 5 is J. 6 is A. 7 is C as in Charlie. 8 is D. 9 is I. 10 is K. 11 is G. 12 is B as in boy. Do you need any of that repeated? Okay, Ashley, are you okay? Okay. All right, page 341. Using your knowledge, letter A, the normal cranial nerve function. Okay, for number 1, hearing the crack of a bat hitting a baseball, that's going to be cranial nerve 8, VIII. Number 2, smelling the dinner cooking, that's cranial nerve 1. 3, being dizzy after going on a tilt-a-whirl, that's cranial nerve 8, VIII. Number 4, lifting your shoulders while doing warm-up exercises, that's Roman numeral XI, that's cranial nerve 11. Number 5, chewing a tender steak, that's going to involve 2 cranial nerves, 5, so that would be Roman numeral V, and XII for 12, so 5 and 12. Number 6, swallowing the tender steak is going to involve 3 cranial nerves. Roman numerals IX for 9, X for 10, and XII for 12, so 9, 10, and 12. Number 7, salivation and crying. Those involve 2 nerves. Roman numeral 7, VII, and Roman numeral 9, IX. Now, Roman numeral 9, cranial nerve 9, is only involved in the salivation, not the crying, just the salivation. Number 8, reading a book is going to involve 4 cranial nerves, 2, 3, 4, and 6. 2, 3, 4, and 6, which as Roman numerals would be II, III, IV, VI. Number 9, seeing a sunset. That's going to be Roman numeral 2, II, so cranial nerve 2. 10, looking cross-eyed is cranial nerve 3, so III is the Roman numeral. 11 is smiling, cranial nerve 7, that will be VII. 12, moving your head from side to side, would be cranial nerve 11, XI. Number 13, feeling a hot curling iron touch your forehead. That's going to be Roman numeral 5, V. Number 14, decreased heart rate is going to be Roman numeral X for cranial nerve 10. Number 15, rolling your eyes. That's going to involve 3 nerves, 3, 4, and 6. So the Roman numerals would be III, IV, and VI. Now, all of the cranial nerves are involved with the digestive system in some way, starting with the mouth. Wow, that's a lot of them. So there's going to be 5 of them. Roman numeral 5, 7, 9, 10, and 12. 5, 7, 9, 10, and 12. Which as Roman numerals would be V, VII, IX, X, and XII. The last one, 17, speaking, talking to someone, 3 nerves. VII, so 7, X, which is 10, and XII, which is 12. That was quite a lot there. Do you need any of that repeated? Okay, the last group here on page 342, letter B, cranial nerve injury. Write the name of the cranial nerves involved. First one, number 18, injury to the cranial nerve causing Bell's palsy. Okay, that would be cranial nerve. That would be the facial cranial nerve. Number 19, injury to this cranial nerve causes a loss of taste sensation, decreased salivation, and difficulty in swallowing. That would be the glossopharyngeal nerve, the glossopharyngeal nerve. And 20, injury to the cranial nerve causes paralysis of the vocal cords. Okay, that would be the vagus nerve, the vagus nerve. Okay. All right, gals, any of that you need repeated? Okay. Is there anything else that I can help you with today before you go off and take that lab exam?
Nope, that's everything. Thank you.
Okay, Suzette. All right then, good luck to you, and we'll see you on Wednesday, okay?
Okay.